Now moving on into the hypothesis of the paper, well, the authors hypothesize that it is possible to engineer for brain-specific organoids from iPSCs which resemble human embryonic cortical development in order to model Zika exposure. To address this hypothesis, the authors first needed to fabricate a new bioreactor which allow low-cost fabrication of organoids. Then they establish a new differentiation protocol for brain-specific cells, and then they needed to validate the organoid models in order to make sure that they resemble the actual tissue in the developing brain. At the end, they expose the organoids to the Zika virus to evaluate its infection mechanism. Okay, so let's move on to the results of the study. So here we have the design of the bioreactor, the miniaturized bioreactor. So it was adapted to fit a commercial 12-well plate. Here we can observe the spinning shaft that fitted into the wells. Those were connected to a gear here, and these gears were finally connected to a motor. So all the shaft and gears were spinning at the same velocity. This allows for a low cost and comparison of large numbers of conditions in parallel. So it was easier for the researchers to optimize the differentiation protocol. So here we can see this exact model. It was just for one 12-well plate, and this is a BS model that allows for the insertion of several 12-well plates. Here we see 3 different. So they optimize this bioreactor to make the differentiation protocol optimization easier for them. This is the differentiation protocol that the authors proposed. They started with induced pluripotent stem cells on day 0. After that, they embedded the cells into matrigel and then regulate some of the specific pathways in order to induce these embryo bodies to a specific brain region. So they regulate the Wnt pathway here. After 14 days in matrigel, they transfer these organoids into the bioreactor, which they call spin omega. Then they introduce different drugs. Then they introduce different drugs into the culture media at different time points in order to regulate the different pathways that will lead the stem cells into forebrain cells. So we can see here that the organization and the cell composition of these organoids on the very first days. If we will take a slice of these organoids, we will see something very simple, mostly composed of pluripotent stem cells. But as long as the process continues in time, the tissue starts to gain more complexity. The stem cells start to differentiate into another types of cells, and they start to separate as well. So by day 14, the researchers took a slice of these organoids, and they observed very interesting characteristics here. So first they observe that these organoids are well defined, that are structured, and they resemble neural tubes, in which we have these like empty spaces in the very center, just like this. These organoids on day 14 highly express beta-catenin, and PKC lambda. These two are adherent junction markers, which means that the cells can communicate with each other within the organoid. They were also highly expressed in nestin and SOX2, which we know that are markers of stem cells, and they were also highly expressive of this marker PH3, which is a proliferation marker. So in combination, all these markers show us that on day 14 the organoids were mainly formed of PSCs. They were proliferating and they were growing. So by day 63, they also took another slice of these organoids, and they observed this other structure. You can see how this structure is also separate in several regions. So in the innermost layer, we have SOX2. We have a high expression of SOX2 here. Here. So this means that in the very core of the organoids, we still have, or they still have these stem cells. On the second layer, they have these cells expressing TBR2, which is an intermediate progenitor cell marker. So this means that the cells in here, the ones that are shown in blue or here, those cells start to differentiate. And finally, in the outermost layer, we can see cells that are expressing CTIP2. This is a neuron marker, so we can see how the cells are differentiating from the very center of the organoid to the outermost layer, which is very similar to what happens in the forebrain and in the cortical development. On the innermost layer here, we will have these stem cells or pluripotent or multipotent stem cells, and then we will have committed neurons. So by day 48, the organoids were very well defined. You can see here again in the very core of the organoids we have a high expression of SOX2, and in the outermost layer we have high expression for neuron markers. So again, the cells kept this organization just as in the cortical brain. Now, to evaluate if this protocol actually worked, the researchers compare the protocol here to other protocols that already existed for brain-like tissue or brain-like organoids. So let's remember that they were looking for the cells to go into this phase. So from forebrain to cerebrum. So first we can observe for the forebrain protocol that the structures are very well organized. We can see this stratification or these layers forming. For the intrinsic protocol, there is not much structure here. And we can see that by day 14, the organoids fabricated with the forebrain protocol were almost exclusively expressing forebrain-specific progenitor markers that are PAX6 and O2X2. So we can see that all the cells in here are expressing either this one or this one, or both. And for the intrinsic protocol, we can see that not all the cells, these for example, this group and this group, are not expressing either PAX6 or OTX2. So this confirms that the proposed protocol is working, that the organization of the organoids is high, just as in the cortical development. They also measure the cells. They also measure the thickness of the layer of stem cells and the thickness of the neuronal layer in the organoids, and the organoids formed with the intrinsic protocol. And we can see here that the organoids fabricated with the foreign protocol have a high thickness in this BC layer, which is a layer that is rich in stem cells, compared to the organoids fabricated with the intrinsic protocol. So this means that the organoids of the foreign protocol have the capacity to keep proliferating and keep differentiating and keep forming this very complex layer structure. Here we can observe in this graphic that the percentage of neural tube-like structure markers were significantly higher for PAX6 and OTX2 for the organoids formed with the foreign protocol again. So these cells, the cells in these organoids were probably starting to differentiate in another cells that are not native from the neural tube. Okay, so they kept this going until day 28, and by day 28 they also observed this really nice, really highly organized structure with the stem cells in the innermost layer, and then neurons, committed neurons or progenitor cell neurons in the outermost layers. So we can see here, this is for stem cells, this is for progenitor cells, and this is for committed neurons. We can observe in the intrinsic protocol that even when there are a lot of stem cells here, they are not organized as in a cortical structure, and there are almost none committed neuron in here. So we can say that this intrinsic protocol actually doesn't work for differentiation of neurons. They also measure the thickness of the BC layer by day 18, and this is even when there is not that much of a difference in thickness, we can see that these are more homogeneous between different samples. They are more alike in different samples and different clones.